Okay, so firstly I would like to extend a warm welcome to you all this evening. We appreciate you coming out in the rain. Unfortunately we've had our summer, I think, but there you go. So for some of you who don't know, I'm Sharon Huws. I'm the Interim Director for the Institute for Global Food Security. And I have the great pleasure this evening of introducing a colleague and a friend that we know very well. So, and that of course is Ermias Kebreab, who is a professor in the animal science area in UC Davis in California. So just to give you a little bit of background about Ermias, started life in East Africa, where he did his undergraduate studies. But since, for the past 15 or so years, has been based in UC Davis, California. Many of you will know Ermias, you will know his work, and that's one of the reasons why you're here this evening. Ermias is absolutely leading in the area of feed additives to reduce methane emissions. And of course, that is something that we work on extensively on this island. But many of you have heard me talk extensively before about feed additives, and I think it is refreshing to have Ermias here tonight to give his perspective on what's happening in California. So I think that is enough from me, and over to you, Ermias. Thank you.
Good evening, everybody. And again, as Sharon said, thank you for coming out in the rain, and thank you for coming out during the bank holiday and in the evening as well. So I appreciate this quite a nice crowd gathering and talking about enteric methane emissions. Let's see if I can work this out. Okay. All right, so I like to start with this sort of overall picture of the environmental impact of not just agriculture, but in general of human activity. Right? So you can think of it as we have a space, we have a safe operating space where the Earth system will be in balance. So the middle part there, the green part there, is basically our safe operating space. What that means is that in the Holocene era, when the Earth system were in balance, that would be where we would expect to be. So there was methane emissions, there were other type of emissions and pollution happening at the time as well, but the Earth system was in balance, so that all of that was basically put into a way that the net primary production was happening. So here you can see that in some areas we are exceeding the what's called the planetary boundaries, particularly in the areas of climate change, but most importantly in biodiversity. So the red zone means that we are really exceeding the planetary boundary. So in biodiversity and also in nitrogen and phosphorus excretion that's coming out, that's where we have the majority impact. In 6 out of the 9 planetary boundaries we have already exceeded. And so today, I won't be able to talk about everything. This will take a long time to cover. So I'll be mostly talking about the climate change aspects. And then from the climate change aspects, I'm only talking about the radiative forcing, which is on methane and not on the carbon dioxide. As you know, carbon dioxide for livestock systems is kind of, agriculture is like a wash, because you have carbon dioxide as being sequestered by the plants that will be fed to animals, and then they respire that. So now then, looking into the methane, so where is the greenhouse gas coming from? So if you look at all economic activities around the world, we probably have about 56 gigatons of carbon dioxide equivalent coming out from all sectors of the economy, and agriculture is about 10.4 gigatons of greenhouse gas emissions. And if you drill down into agriculture, where is it coming from? The biggest part is the land use change. In a lot of places, we can't really do much about it. Where I come from in the US, we've cut our forests a long time ago, so there's not much land use change happening, but in other parts of the world, there's still going on. So the next from that is enteric fermentation. So the enteric emissions piece in agriculture is quite big. So next to the land use change, the enteric fermentation is quite, has a big contributor to the emissions. And that's why we're focusing on that and trying to figure out how we can reduce the impact of livestock systems in this case and reduce enteric methane emissions. So why are we worried about methane? Well, methane, if compared to the before the industrialization, we have now about 200% more. So we have really over doubled the concentration of methane in the atmosphere. And what does that mean? Well, methane is, depending on how long you consider the impact, over 20 years it will be about 86 or something like that, and over 100 years it will be about 28 or 27 point something. It will be the impact. But methane lives in the atmosphere for 10 to 12 years. So while it is up there, it's very, very effective in trapping heat so that it is a potent greenhouse gas. So that's why we are interested in that. And if you calculate how much warming has happened since the 1800s, it's about 1.1 degrees centigrade. And almost half of that, 0.5 degrees centigrade, is because of methane. So although methane is not as much as carbon dioxide, its effect is very much outside. So if you are able to reduce methane emissions by about by 30% or so by 2040, we can save the 0.3 degrees centigrade warming to the environment. So considering that we want to stay under 1.5 or 2 degrees centigrade below the 1800s, we don't have much room left. We are already 1.1 above, and so we only have 0.3 to 0.4 to get to the 0.5. So if we are able to reduce methane emissions, then there is a huge amount of reduction that could happen. And in the short term, it will help us slow down the warming that's going to happen. So that's a lot of focus on this. So since the 2021, I think it was the COP meeting in Glasgow, there was a renewed emphasis on methane. Before that, there wasn't much really focused on that. It was mostly about carbon dioxide and its effect, but IPCC really put a lot of effort in educating people, and I think it's about that time, 2021, where the global methane pledge was signed. About 100 countries signed. That was started with the US and EU, and then it spread to other countries, and now in the last COP, I think it went to 150 countries have signed it. And if all these countries actually did what they said they were going to be doing, then there will be about 0.2 degrees centigrade savings from that action as well. And then through that agreement, now there is a new sort of organization, the Global Methane Hub, who is funding a lot of those projects to try to limit the amount of methane that's coming, not just from agriculture, but from other sectors as well. So the FAO put this out last year, I think it was 2023. And basically what it shows here is that, so the first column is, this is the base year and how much emissions are happening at the moment. And then the next one to it is, if we continue the way we are doing right now, where we are headed. How much more emissions are we going to have? And so it's a huge amount of emissions that we'll have. And then the subsequent columns show, if we do some things, what are the effects of reducing the emissions? So things like productivity increases, for example. So if we increase the productivity of some livestock, we could reduce the amount of methane, at least the methane intensity, by a certain amount. And so they went through the different ways to reduce it. And some of it include rumen manipulation, also looking into improved health and things like that, that I was going to be discussing this evening, but mostly focused on the rumen and how we can target the different species within the rumen to be able to reduce the methane emissions. So basically, this is showing that there are different ways in which we can reduce the emissions. This is not specifically for enteric emissions, but enteric emissions has a big contribution to this. So now, mostly really focusing on some of the mitigation aspects that we can see. So we looked at a few years ago, what are the enteric methane emission solutions that we already have? And so we classified it into different categories. We have the animal feed category, animal and feed management category, and then we have diet formulation, and we have the rumen manipulation as well. I'm not going to go through all of these different things, but the idea was if we implement this, where we will be in the future. And what we found out is that we could stay below 1.5 if we implement some of this by 2030, but by 2050, we are not going to be able to do that. So we need new kind of methodologies, new mitigation options to be able to help us to stay at that 1.5 level by 2050. So I'm going to focus more on the things that are that have been found to be quite effective, and then we'll go through the other methodologies that are upcoming and something that we actually will be able to use in the future. So if you look at in terms of effectiveness, so this graph here shows different types of additives that are effective in reducing methane emissions. It doesn't say anything about the ease of use and commercialization or anything like that, just in terms of effectiveness. You see there the seaweed, different seaweed species have the potential to reduce emissions quite substantially. Including lipids or fatty acids also has the potential to reduce emissions. Bovaer or 3-NOP has the same effect. And then we have secondary plant compounds, particularly tannins, oregano, essential oils, and things like that. So what I'm going to do is go through those one by one and see where we are. There's been a lot of experiments that have been done, recently published experiments, meta-analysis that have been done. So just to sort of update you on exactly on what's going on and what kind of scientific evidence do we have right now if you're going to use some of those technologies. So the seaweed story, I guess, it was kind of began around 2016 or so in Australia, where they were screening a lot of different type of seaweed from the coast of Australia, and they found that in particular one species, Asparagopsis species, showed the greatest reduction in emissions, and it got us really interested in that. I think 2017 or so, we contacted the colleagues in Australia, and then we were able to get some samples from them, and we did this in vitro test, and we found the same kind of result as well. And we thought, well, we should really do this in animals. So at that point, there were no experiments done in large ruminants, so we didn't really know what was the inclusion level. So we did a small experiment, and then we tried it out at 0.5% and 1%. And at the time, the only species that we could get was Asparagopsis armata. We actually sent divers off the coast of Australia to go down and collect this. So it wasn't a very homogeneous kind of thing. And when they brought it up, there's a lot of rocks and corals and everything else. So we had to process it, and they freeze dried it, and they shipped it to California. And we did this first experiment in dairy cattle, and we found a very substantial reduction in emissions. At 1% level, I think it was about 60% reduction in emissions. But at the same time, we saw a reduction in feed intake also. The cows, especially at 1%, they did not like the seaweed very well. We had a huge challenge in trying to get them to eat. So 1% was probably was too much for at least for the dairy cattle. So the next experiment we did, we used a different species, Asparagopsis taxiformis, which had a higher content of the bioactive, and that seemed to work much better. And then once we had a sort of a good result with the beef cattle, so we did this in beef cattle, we reduced it up to 0.5% of the seaweed. We have about 80% reduction in emissions. And then we thought, okay, so how can we translate this into grazing systems? So we were able to get a rancher. Some of you might have heard of. Rupert Murdoch had a big farm in Montana. So we worked with his farm in Montana to put this together, and they helped us set up this green feed. This is a double hopper green feed. So basically, you have a large box divided into two, and depending on which animal is coming, it will drop either the control pellet or the treatment pellet. So we pelletize the Asparagopsis, and that was actually quite a big learning curve for us because again, it wasn't pelletized before, and we cannot do a normal pelletization where you warm it up, or heat pelletization would not be good because you will lose all the active ingredient. So we had to do cold press kind of thing. So it took us quite a bit of time to try to come up with the product that we can actually use. And once we used that, the palatability issue was still there because now you're actually concentrating it to give it as pellets. So we had to experiment with a lot of flavor enhancers. And finally, we got into a place where the animals were able to eat it. And so we started feeding this. So we did a trial. We wanted to do a trial for 6 months because that would give us a nice data, but because of all these issues in terms of palatability and trying to figure out how to pelletize this, took so much time. So in the end, we ended up doing a 10-week trial. And what we saw there was that there was a substantial reduction in emissions. So this is a totally grazing system. And we saw quite a big variety. If animals eat it, there is a reduction. If they don't, then there isn't. So some animals reduce 80%, 90%, others 10%, 20%. So on average, it was close to 40%, but the variability is there is really to do with the bromoform intake. So we try to show here, as you increase the bromoform intake, you see the reduction there as well. So it's really a linear kind of relationship between how much they are eating and how much reduction is happening as well. And then after about 5-6 weeks, so the intake level of the bromoform itself went down, and so that you see that the things coming up again. So the reason it's coming up is not because there's any kind of adaptation going on, but it's because they're not consuming enough of the material that we were providing them. And then we looked at sort of the effect on average daily gain. Obviously, we only had about 20 animals or so we can't really pick up small differences in average daily gain. But at least we can say that there was no significant difference between the control and the treatment. So if we had more animals, if you continued this for a longer period of time, you may be able to pick up some differences, but at least we can comfortably say that there was no significant difference between two. So it didn't affect average daily gain, but we saw quite a big reduction in emissions. Now, you can ask if you know the active ingredient, which we know is mostly going to be bromoform, although some paper recently was saying that bromoform is maybe 60%. There are others that are doing that. I mean, that's debatable because that was done in vitro. I haven't seen that comparison in vivo. But if you know the active ingredient in bromoform, why don't you just synthesize that? And we know how to synthesize bromoform and use that instead of going into trouble of cultivating seaweed and doing all that. Well, there are a couple of companies that are doing that. One based in California and another one based in Australia. And we're interested in working with them to see if that would actually happen. And so we did this experiment. It was published maybe a month or two ago. And what we saw, so what we did was we had two formulations, a powder formulation and then an oil formulation. So basically the bromoform suspended in oil or the bromoform in a powder format, stabilized bromoform in a powder format. And when we fed this to beef cattle, what we found was that there was about 95% reduction in emissions the oil, but not on the powder. So the powder, we had a bit of an issue there, unfortunately, because we thought that we were given the same amount of bromoform in oil and powder format, but when we analyzed it, we found that the powder format actually had like 2 milligram per kilogram of bromoform, and so we contacted the ruminate to say, hey, these are completely different ones. The oil has like 32, that's what we were expecting, but the powder had much less. So they gave us a second batch. Unfortunately, that batch also had only 17 milligrams per kilogram. So I wouldn't, I don't believe too much on the powder issue. I think if we had a similar level, we probably see a reduction as well. But the oil, there was no, I think that was very clear. The bromoform, just pure bromoform suspended in oil, you feed the animals, this is what we got. And there was no effect on intake. So that's the advantage of having this synthetic bromoform, was that at that level, at 32 milligram per kilogram, there was no effect on intake. And we also hadn't seen any kind of change in behavior or in average daily gain as well. So I think synthetic bromoform would work, but you have to be very careful in terms of maintaining that bromoform until it gets into the rumen, because it's a volatile compound. If you don't stabilize it and make it available in the rumen, then you will lose that, and you're not going to get the mitigation that you're getting. But this is, a lot of companies are getting really interested. There are a lot of chemical companies that produce bromoform as just normal business. Now they are thinking, well, maybe we could do that. We could do the same as well. Well, Ruminate has a patented, so they have a process that they patented, which means that others could not use the same process. But that's not going to stop a lot of people. So they are trying to do the same thing. And the next couple of years, I won't be surprised that there are more startups or established companies that are producing bromoform, especially if there is regulatory approval for ruminate. Then there could be more of this coming up. And Ruminate has a lot of backing. They've raised quite a lot of funds, and they strongly believe that this will be approved in many countries. So we'll see. I mean, in Australia they can do it, but that's the only country they can do it so far. But I see a challenging regulatory path, but I may be proven wrong. So it's not just about the bromoform, but most of the halogenated compounds would have similar impact as well. So this is work done in Denmark, where they looked at iodoform instead of bromoform, and they've shown the same thing as well. This is a dose response that you can see that at the top is the control, and then as you increase the concentration of iodoform, you see the reduction in methane as well. So it doesn't work as well as bromoform, but it's just to prove that halogenated compounds would elicit the same way reduction in methane emissions as well. So I don't know if the Danish team are going to continue with this line of research, but the bromoform, it's clear that there's quite a lot of companies interested in pursuing this. Very recently, we put together a meta-analysis on bromoform. So by now, there were about 14 papers published, and they had about 39 treatment means that we were able to collect, both from synthetic bromoform. There were three papers on synthetic bromoform, and the rest were a mixture of taxiformis and armata. And what we've seen is that at the average dose, so the average dose for this work was about 28 milligram of bromoform per kilogram of dry matter intake, and we saw about 47% reduction. So that's the average. But then we saw a big impact of the substrate as well. So depending on the NDF and starch content, then that could vary quite a bit. So NDF, if you have more NDF, more fiber, you have less effectiveness of bromoform. If you have more starch, then you have a better effectiveness as well in this case. So this is not published yet. It's only on a preprint, but it's going through the review process at the moment. So the next one I want to talk about is, I'm sure most of you have heard of Bovaer or 3-NOP. This is the most studied feed additive that we have right now. I think there are at least 60 studies that have been conducted there. And in 2023, we published a meta-analysis on dairy. And in that meta-analysis, we've shown that on average, the reduction is about 30%. And then right now, we are working on very similar work but for beef. So we will have a meta-analysis on beef very shortly. I'm hoping by end of June, we will submit that paper on meta-analysis for beef as well. We've seen it also, the impact of NDF on the effectiveness of Bovaer. So again, similar to seaweed, you increase the NDF, the effectiveness is lower as well. But a lot of this work was done 3-4 months. The typical research that we do is about 3-4 months. So there's been questions about what happens when you continuously do this for a year, throughout lactation or throughout the life stage of beef cattle. So this is the only work that I can find in the literature where they've fed it for the full year, so the full lactation. And here we can see that the effectiveness was throughout the whole year. It kind of changed a little bit from time to time, which again, this is what we have predicted, just if you look at the equations that was developed, because when you have better feed quality, lower NDF, sort of at the beginning of lactation, the impact was higher. So you see much more reductions, I think about 25% or so reduction that you will see when the NDF level is lower, and then the feed quality in general is higher. And then when you go through the lactation and you go to a dry stage where the quality of the feed goes down, then the effectiveness also goes down. But what's really interesting is that when they go to the next lactation and then you change the diet to become a higher quality diet, then the effectiveness increases again. So I think this is a very encouraging result that you see that there is a persistence in terms of the reduction of emissions happening throughout the lactation, and not just one lactation, but going into the second lactation, you will see that continuing impact as well. And I know there are a number of other work that's going on. I think there's one in Penn State on this. At UC Davis, they're also just starting to do this work for a longer term as well. So this kind of data is very helpful to see when you're doing it for a longer period of time, what are the expected reductions? Because sometimes doing it just for 3 months may not reflect what will happen in a commercial situation. So I think this is pretty good data, and then more data should be coming up soon as well. One of the really interesting things that's start to happen is thinking about the early life of the animal, and can you reprogram the rumen in early life? I think this is a really interesting question where you may be able to intervene right at the beginning, and then the animal would then continue to have lower emissions throughout its life. One of the work that was done recently showing that they tried to give, this is also in using 3-NOP. 3-NOP towards the end of, just before the cow gave birth, and then continue with the calves, or you introduce it at the calf level. So they looked at it both, giving it to the dam and then the calves, or just to the calves. And what they found was that the first set of graph there, they didn't see any significant difference when they gave it both to the dam and the calf. But when they gave it just to the calf, this seems to work well, and you can see that there was a continued reduction in emissions after they gave it to the calves until it's weaned, and then they stopped the treatment, and it continued to have a lower emission. So you can speculate, for a number of things, and people who know about microbiome a lot more than me, like Sharon, will be able to speculate why that's happening. But it's really interesting to see that you can actually effectively reprogram the rumen to have a lower emissions once you stop the treatment. So this is work that we are doing at UC Davis as well. And in this project, this is part of a much bigger project. What we were doing is that we have 3 treatments. One is the control, and the other one is we start the treatments right after they are born. So the first day they are born, we start the treatment. This is with seaweed. So this is oil-based seaweed, and we introduce that, and then we give them until they are weaned. Once they are weaned, we stop the treatment, and then we continue to measure the emissions. And then we have a third treatment where we start after they are weaned, and then we continue all the way until slaughter. This is just the first 7 months, and because we don't have a big dairy, we were enrolling calves as they were born. And we were using bull calves, and we used sex semen, so we were just praying that there was, we can get bull calves. Every time a bull calf is born, then we put it into the experiment. So it took us about 6 months to get all the 24 animals that we need. So these are the averages of maybe 6 or 7 animals. So next year we will have a much better picture. But still, the picture is becoming clear here, where when we have the calves and then we stopped it after weaning, at least for the next 4 months, there were still lower emissions, comparable to the ones that we were giving them on a daily basis. So this is really amazing to see that, those on the dotted line, there was no additive. They were given additive until they are weaned for the first 2 months, and then that was it. And then when we continue measuring them, we can see that about 30% reduction continuously until about 4 months, and then there was at the 5th month there's a peak, and then it's go down. As I said, because there are only 4 or 5 animals here, the data is going to be variable. But once we get the 24 animals, then hopefully it will be stable, and then we can actually see that. And for those of you who are coming to GGAA, we will have an updated results on that. So the postdoc that's working on this will give a presentation on this. Moving on to other mitigation, we also just finished an analysis on dietary lipids. So basically, the US government wanted, not this government, the previous government, wanted to know about what are the mitigation options that we should recommend to farmers. So we were working this for the last year, not just on enteric. So we had a big team of people. So I was leading the enteric part. There were other people working on manure management, forestry, on carbon sequestration, and all that kind of stuff. So on the enteric side, what we are recommending is 3 things. One is bovine, because now in the US it's approved, so that can be used. And second is lipids, and third is tannins. So we were interested in giving them scientific based advice. So we needed to do a meta-analysis of all of those things. So for 3-NOP, we already done it, and then the beef is coming. For lipids, there were actually a lot of meta-analysis that was done, but 13, I think we found there 13 meta-analysis that was done. So what we did was we went to a second-order meta-analysis. So this is a meta-analysis of a meta-analysis to try to figure out what's the impact of feeding lipids in reduction emissions. And the other thing that we're interested in is depending on your baseline, it changes. So if your diet already has 2%, then what would be the impact when you add 1 or 2 or 3 percentage points? If your diet is already at 5%, there's not much room to do that, because you cannot keep adding. After 6, maybe 7%, that's it. Then you're going to have other problems. So so in this work, what we're trying to give guidance, what's the lipid content of your diet first. Once you know the lipid content of your diet, then you can calculate, if you add 1 percentage point, then usually I think it's about 13, 12%, depending on where you are. The less lipid you have in your diet, the more mitigation you're going to see later on. So we are just finished this work, so hopefully by end of June again, we will try to make it available at least as a preprint, so that people can have a look at that. So the next thing I wanted to add is on secondary plant compounds, particularly tannins. In California, we have a big wine industry, and once you've squeezed out all the juices, there's a lot of pomace that's left. And at the moment, it's not really used much. And some of it has been used for as a fertilizer kind of thing. You compost it for a little bit, but there's not much that's been done because there's limitation on how much of that you can compost. So a lot of it just goes into the landfill. So the idea is grape hummus contains a lot of tannins. It contains some lipids as well. So why can't we feed it to dairy cattle? And there is some evidence from Australia also that have done this before, and they've seen some positive actives. So the dairy farmers of California, they asked us to do this work, or they supported us to do this work. And what we did was, it's a fairly simple thing. We collected the grape hummus from the winery, which is not far from Davis, and we looked at the tannin contents and the polyphenols in different ways. One is just directly the wet hummus, and then one is the dried it first, and then we looked at it, and the other one is we dried it and then we ground it into a powder. And what we found is that the wet unprocessed had the highest amount of tannins and polyphenols. So that's what we used. So we just basically literally drove into the winery, put it in the truck, and then bring it to our dairy and feed to animals. So we have a 10% and 15% levels, and we saw a reduction in methane emissions, but the reduction in methane emissions is mostly related to a reduction of dry matter intake as well. So there was a little bit of lower dry matter intake and then lower emissions, but the milk production was the same. So there was a better feed efficiency there. We didn't see a drop in milk production, and then there's a little bit of less dry matter intake. So in general, I think it's a good news for farmers, and the quality of milk also improved. So we worked with colleagues in food science that analyzed the fatty acid content of the milk, and they found that it has higher unsaturated fatty acids there, which was better for human health. So it has a better quality milk, and then reduced emissions and better feed efficiency as well. And so we are interested in continuing this line of work. We're going to be doing some more work with olives as well. So we have a lot of olive production. The same thing there's a lot of olive hummus. So the olive industry is interested in this kind of work as well. So we will do this, and then we'll repeat the result that we have. At 15% is probably too much, and so we're going to be probably working around that 10% value. But at the moment, if we use that even at 10%, there's not enough grape hummus for all the dairy cattle we have in California. We have 1.8 million, there's not enough. So we're trying to figure out what would be the right amount, and it has to be, if it is wet hummus, that's good, then the dairies have to be fairly close to the wineries, which, there's quite a lot of dairies that would be able to take advantage of this. One that's we just finished is looking into almond hulls. Almond hulls, again, we produce a lot of almond hulls in California. We do use already the almond hulls, but the idea was if you ferment the almond hulls and get the fermentation process going, if you feed those, what can you do? Can you reduce emissions because you already started the fermentation process? So when we tried that at 5 and 10%, we saw a little bit reduction, a little bit but significant reduction in emissions, about 10% reduction in emissions when you give fermented almond hulls. And the other good thing is that we also saw a little bit of increase in milk yield. So even at 5%, there was a better milk yield as well. So what we're going to be doing in this is moving forward is to try to figure out, is the cost, would you be able to pay for fermenting the almond hulls first and get this benefit? Is it beneficial? Or you can just feed it the almond hulls the way that people are doing right now. And so we're going to do some cost-benefit analysis and see if this is something that we will recommend for farmers to use. The other thing that as I said earlier, there are 3 things that we recommended for U.S. farmers. The third one being tannin-based additives. And so we did a meta-analysis also on tannins. And on this one, we were very specific in terms of what we wanted to include in the meta-analysis. It has to be something that you can physically add into the diet. We're not when we're not looking into tannins, like, if you give plants that have higher tannin content, or like even grape pomace and things like that, that is not the idea. The idea is something that you can actually supplement to the animals and then see an effect in emissions. And what we saw was that close to 10%, actually, this was an older version when we do the reanalysis, the methane production and yield was about 10% reduction. So you're just adding the specific tannins, you could get up to 10% reduction in emissions, which I think is pretty good. And there was some other effects of tannins, and a lot of people use it, sort of anti-parasitic kind of uses, and so there's some importance in immunological importance and things like that. So if there are other co-benefits, then you might as well use it and reduce methane emissions as well. Essential oils, we really had a big hope for this. Agaline was touted to be reduced methane emissions. There was even a meta-analysis that was done that showed that reduces emissions by 8-9%. Unfortunately, that meta-analysis included a lot of sort of in vitro kind of work. It wasn't all in vivo. A lot of the studies in that meta-analysis were not published as well, so you can't really independently verify what was all happening. In any case, we did work. This is one of the challenging work that we've done, because this is we did it in farmers' fields because we wanted to try to figure out, a small differences, about 10% or less, maybe 8 to 10% differences is our target. So we had to use 72 animals on farm. So very involved kind of work with 3 green feeds, 3 double hopper green feeds. And what we found is that unfortunately it didn't really work. So there was only 1 week out of the 10 where we saw those milk production was increasing, and then the methane was a little bit lower, so it came out to be significant. But really, for the whole duration, if you look at the whole duration, there was no significant differences in there. So yeah, unfortunately, it didn't work for us. But again, there's more and more work coming out that also show the same thing. Like a few people have tried it. I was in Denmark last month, and people tell me they also tried it there. It didn't work. People in Switzerland tried it. It didn't work. So this is not an outlier. But there was this idea. There's a company called Methane, and basically what they do is that they look at the microbiome, and through their some sort of AI analysis, they can identify the microbiomes that so the animals with specific microbiomes, if you feed this agaline, would work. In others, it doesn't work. So they looked at 13 different farms, and they were able to predict. So it reduces emissions in some of them, and in some of them it actually increases. So there was from 8% increase to 20% decrease. And I mean, to me, it's kind of, if you look at it's like a bell curve. You have from 8 to minus 20, then the middle is going to be like 0. So I don't know. I haven't really seen exactly how they're the work that they're doing is applied, but the idea is that we take samples from the rumen, you look at the microbiome, and you can identify whether this feed additive would work in that microbiome or not. So I think the idea is great, but I think there's a long way to go. So this is a nice segue to talk about microbiomes. So there's microbial engineering. In order to be able to do something with microbial engineering, there's a lot of work that's happening right here in Queen's University is leading this work. This is a very international work where there's a lot of partners around the world to try to really understand the microbiomes, the rumen microbiome, so that using this, then you can do quite a lot of different things. It helps you in terms of mitigation. So we recently started this new idea in microbiome engineering. using the CRISPR-Cas system. And this is an initiative from the TED Audacious project. So TED Audacious, basically, they will fund projects. It's kind of high risk and high return kind of work. So this is something that no industry will fund because it's just too risky. But with this kind of support, basically people would say, yes, we think this would work. If it doesn't work, it's okay, but at least we're trying. But within this work, we will be able to collect quite a lot of a big sample. I think Sharon, you will like this because we're taking samples every month from those 24 animals that I showed you earlier, from birth to slaughter, and all of that data is going to be sequenced to death. I mean, there's going to be a lot of sequencing happening at UC Berkeley each month to try to understand what is changing. So relative to the control, when you give additives, how is it changing? And when we were doing this early programming, that we're seeing there was a reduction. So why is that happening? So trying to understand what's the microbiome look like when you have this early programming. So that's really exciting work. Unfortunately, it hasn't started yet. So the samples are all at UC Berkeley, and now we have all the 24 animals. They're going to start working on it and try to understand what's the difference in the microbiomes for animals that have reduced emissions. The reason we're doing this is we wanted to understand what are the microbes that we make sure that are there when we do this the editing of the genes. So the idea is it's really the truly enabling technology is this effort of the CRISPR-Cas system, where you can try to identify the genes that are responsible for methanogenesis, and you go there very specifically and cut out those or edit those genes and be able, in theory, if you do that, then you have very little to no methane emissions happening. So the first step is to really figure out what are the editable microbes within the system that are responsible for methanogenesis. And then once you identify those editable microbes, then you edit out the specific ones, and so that you're not going to get methane emissions. This is, again, as I said, it's high-risk stuff. There's a lot of people involved, a lot of teams involved, people who are doing the editing, and people also, once you figure that out, you need to be able to get it into the animal as well. So how do we get it into the animal? Is it intravenous? Is it like a probiotic that the animals would take? Is it better to do it when they are calves, or is it better to do it when they're adults? There's a lot of questions. We have 5 more years to figure that out. But along the way, we're trying to learn more about it. Already what we've learned is that there are microbes that are utilizing hydrogen. So hydrogenase producing microbes are really promoted when you reduce methane. So we are identifying those microbes that are utilizing hydrogen. So the idea we have now is that, well, instead of going after methanogens, maybe we should go with these other microbes and increase their numbers in the rumen, and so that with this with the hydrogenase enzyme, be able to utilize the hydrogen, so that there is more competition, and the byproduct of that is that you have more product. If you use the hydrogen, then you will be able to actually convert it into a usable product, rather than reducing methane, and then some of the methane or some of the hydrogen will just go away. So to capture hydrogen is actually better to promote those microbes that utilize hydrogen. So this is just 3 months ago that we were thinking about this kind of thing. So maybe in another 3 months we might go in a different direction. But that's the beauty of having this kind of support, is that we can really change the way we want it. So we're not really limited where we want to go with this kind of project. There are different ways in which you deliver these technologies. Right now, a lot of the feed additives are given as a mixed with the diet. So the total mixed ration is the ideal way in which a lot of the feed additives have been provided, but obviously it has limitations. Most animals around the world are on pasture, so this really is not going to work a lot for most animals. So there's a lot of work now being done on slow release boluses. So you put this into boluses and take it into the animal, and then it will be released at a predetermined rate. So this could stay from weeks to even up to a year, where you get this amount being released very slowly, and then finally the bolus itself will disintegrate and will basically disappear within the rumen as well. So you don't need to try to take that out, and that will make it almost untenable if you have something like that. So this is on the works. There's a company in Australia or New Zealand that already have done this, have patents on this. So I'm expecting some more work to start on this as well. So this will be very relevant here, where we have more pasture-based production systems. The other way is incorporating with as mineral blocks. The only problem here is that you won't be able to have control on how much they are consuming. So there are technologists in trying to figure out, based on the movement of the head, how much of that is being consumed, so that you can calculate how much emissions you would expect to be reduced. But you won't be able to control how much of that additive is being consumed. So water-soluble additives are another thing that's happening. Ruminants have a water-soluble additive. We were going to test that in Davis. Unfortunately, we didn't get the funding to do that, but they do have that. I think they are doing it in Brazil now. So you'll see more and more work coming up with additives being delivered in different ways. And then the microbial engineering, as I said, we're trying to do it in a way that will just do it once, and then if that works, the next generation will probably have the same microbiome as well. So I think if you do it once, it will continue to reduce emissions. So it's not a great business model, because you can't sell it forever, but it will be very effective if it works. So I'm going to mention some things very, very quickly. The breeding is another thing that's happening. There's teams all over the world. Global Methane Hub put together this big consortium working on breeding. In Canada, there's the Genome Canada that's also working on that. So the idea is that through selective breeding, you can select animals that are inherently low methane emitting. In New Zealand, they have done that with sheep. I think they've gone about 12% reduction over 10 years. And so similarly with dairy cattle, there is also an effort to do that. And the good thing about it is that it's very applicable to pasture-based animals. You don't have to do anything once you have the low-emitting animals. And then it's cumulative and it's permanent as well. So it's very beneficial, but it takes a long time. So in the future, it will be helpful, but in the short term, the big reductions will be in additives. The other thing that I put this very last minute is the vaccines, because I've seen a lot of progress that's happened in relatively short period of time on vaccine. This was tried 20, 25 years ago, like in Australia, but it didn't really work that well. About 8% reduction, and also you have to formulate the vaccines for different animals in different geographies and all that. But now the vaccine technology has improved quite a bit, thanks to COVID. So there's a renewed interest in vaccines. And there are a few companies like Archaea Bio, Pasture Bio, and others that are working in this area. And where they are at the moment is that they are able to have a stable antigen production. So they have compounds that would give you a consistent antigen against methanogens. So that's been shown well in vitro. Now they're just getting into the in vivo space to make sure that this works in vivo. So there's a number of questions that will be asked in vivo. So this is the work that we will be starting very soon as well with a couple of those companies. They've done some preliminary trials. And last week there was a summit in Davis, and in that summit they presented about 13-15% reduction in emissions in a preliminary trial, in vivo trial. So we're going to be doing sort of a properly designed experiment to figure out what would be the expected reduction. What they do is the vaccination and then a booster. So the question is that enough? And for how long is it going to be enough? Is it going to be for a whole month, or is it going to be a month? For how often do you need to do the boosters in order to get that level of reduction? So we had a meeting with the legislator or the regulators in the in California state, and that's what their question was. So they were saying for us to be able to implement this, we need to know experimentally how long is this going to work. So if your experiment is 3 months, then we'll give you so the approval for 3 months. If you show us for a year, then we'll give you for a year. So there's a lot of work to be done, but we are in a stage where things are going to move into the in vivo space, so, which is pretty exciting. So towards sort of finalizing, the one thing I want to comment on is that a lot of the strategies that I showed you will be mostly in Western countries, but a lot of the methane, about 80% of methane is coming from low and middle income countries. And so the strategy to do that, to reduce methane emissions from those countries, is not going to be the same as all this stuff that I have been showing you earlier. So what we need to do in those countries is really improve their productivity and reduce the methane intensity by improving the productivity. So in this space, we're working with 13 countries at the moment. And what we're doing is we are developing the nutrient requirement of animals. So a lot of the nutrient requirement work was done for temperate cattle, particularly Holstein and Jerseys and all that, but tropical cattle, we don't have as much data. So that's what we're doing, trying to develop a nutrient formulation really for tropical cattle. At the same time, we're trying to get a low-cost way to analyze feed. So one of the problems we have in tropical countries is that there are no labs that will analyze your feed and then tell you what the quality is. So there's an infrared-based technique that we are using. It's going to be handheld. And the other really innovative stuff that we're trying to do is using your cell phone and getting some nutrient quality from your cell phone. So the way we're doing it is every time we collect feed data, we have this high-resolution camera, and that high-resolution camera will take a picture, and then we have a wet chemistry analysis done on the same sample. And then through repeated sampling, then there will be a model that will be trained on this camera. So they will have this comparison between the wet chemistry and this high-resolution camera. In the end, we can translate it into something that you can use your cell phone, take a picture, and then it will tell you what kind of, what's the crude protein, NDF, at least the major ones through that way. And that then will linked with a software that you can just upload into the software, and then you can get a ration formulation done through that way. And because we work in a lot of different countries, obviously it has to be in the language that people are speaking. So we are going to move it into what we call the farmer chat. So this is an AI-assisted thing. And what would happen is that people would speak into the software. They will basically say, I have a dairy cow weighing this amount, and right now I'm feeding this, and I have this and this feed available, or you can upload the pictures of the feed you have availability, and then it will do the ration formulation, and then it will come up in the same language that you tell it. It will tell you, well, if you want to get 20 liters a day, then you should be feeding 20% of your this silage or 20% of that, and so that you can get a recommendation. So this is the kind of stuff that we're doing. So right now we are in the phase where we collected 2 seasons from all these countries, and we have all the data available, and we're training for the camera-based nutrient evaluation. So hopefully we'll get there, and we have 2 more years to work on this. So this is the final slide, just to show where we are in terms of inhibitors. We have quite a number of players here. I put Elanco for Bovaer because that was the US, but I guess here I should put DSM. But for seaweed, there's quite a lot of companies. They have Fisher Feeds, Symbrosia, Blue Ocean Barnes, and then synthetic ones like Ruminate coming up. And then you have other feed additives like lipids and byproducts like tannins and other byproducts. Genetic selection is going to be quite important moving forward. And then we have tools like microbial engineering and trying to increase the productivity of animals so that we can reduce the methane intensity, particularly in low and middle income countries. So this work is supported by so many different organizations. I just want to say thank you to everybody that has supported this work, and then thank you for the invitation to come and spend this time with you. Thank you.